Thank you. Hi, everybody. So I let the last people sitting down, taking their meal. Okay, let's go. So thank you very much for attending this session. So it's our Twist Corporate State meeting. So today we will present a few things. So the first presentation, it will be the Twist presentation, meaning that we will introduce the Twist technology, what's behind the magic of the Twist probes for your sequencing. And after this, we have the pleasure to have two speakers. So Mayra Sauer, so is co-leader of the MGZ unit, that's it. And she will introduce a nice workflow, long-read workflow with the Twist product. And after this, we will have also Julie Vendrell, so she works in the CHU of Montpellier in France. You will see that she implements different kind of things, and in my presentation as well, I will show you some HRD things that we did together. But in this case, it will be cell-free DNA workflow, so with a really high coverage with the UMI from Twist. So just to introduce myself, I'm Yann Merlet. So I'm the field application scientist in Twist. So I cover the south of France, of Europe. So I work for Twist for 6 years, and I'm really happy to work there because we have really nice products. So to give you a short introduction of the Twist company, Twist has been founded in 2013. So it's a company that has been established with Emily Leproust, Bill Bake, and Bill Banay. So they all work in the DNA synthesis technology. So our headquarters is based in San Francisco, but as you can see on the map, we have all over the world some offices. So we opened a new factory of the future. It's 10,000 square meters, something like that in Portland in 2022, just to double our capacity of production to support our growth. So all of you know that DNA is quite important in any kind of field of studies or for any kind of product. So it's great because Twist, we make DNA. We make it really well, and with this high quality DNA, we can enter in different field of applications, so chemical, food, therapeutic, diagnostic. Today we will talk about target enrichment, obviously. So you have to know that we are in the capacity of producing more than 5 million of oligo per day, and this is thanks to technology. So just a quick summary of our technology. So our major innovation is to miniaturize the DNA synthesis, the fast-write DNA synthesis. So this has been done on the silicon platform. So this is the size of an iPhone. Okay, on this iPhone, on this silicon platform, we can synthesize DNA. So the traditional chemistry, the phosphoramidite chemistry, which was done on 96 well plate, has some drawback. So the major innovation is the thing that we miniaturize. So miniaturizing this, there are a couple of advantages. The first advantage is the fact that when we miniaturize, we use a less amount of volume of reagent, obviously. So this is great. So meaning that on the silicon platform, we work at a femtomal scale. So this is really great. So if we talk about the carbon footprint, just to give you an idea, the traditional method, it's the equivalent of driving 95 kilometers. Okay? With our silicon platform, it's almost 150 meters. So this is the first advantage. The second advantage, it's the fact that with the silicon platform, we have high throughput. So we multiply by 10,000 our capacity of production compared to the traditional method. And the third one, which is the best for me, it's the fact that on the silicon platform, we have a precise control of the concentration. So this is quite important because when you produce probes uniformly on the GC spectrum, you will have a uniform capture. I will show you exactly what it gives at the end. So yes, on the silicon platform, the idea is to synthesize probes. So it will be single-strand DNA probes. Okay? So, but we keep this GC spectrum really flat, as you can see on the slide. But the drawback of this is when you amplify this, because since we work at a femtomal scale, we need to have more yield, more DNA. So the idea is to amplify this, and the traditional method to amplify this gives a lot of bias. So when you have this kind of bias, you will have a non-uniform capture, and it will lead to a non-uniform sequencing for your data. So that's what I showed you on the top. You will have under-sequence region, over-sequence region. Our goal at Twist is to have to keep this GC spectrum uniformly synthesized or GC spectrum reproduction, let's say. So we keep this after this amplification method. So at the end, we will have a uniform capture on our target region. It will optimize your sequencing throughput, obviously, and reduce your sequencing cost. So our probes are double-strand probes, 120 bare base probes. So after denaturation, you can target the sense and antisense in one synthesis, which is not the case of our competitors. For this, okay, we have nice probes, double-strand probes, but we need a tool. We needed a tool for this. So we set up a tool. We call it the Rapid Design Build Test Learn Cycle tool. It's easy to understand. Our nice bioinformatics expert will design probes with a lot of filters, a lot of tools, and then we will print it on the silicon platform. After printing it, we will QC them, so NGS QC, and then we will see if we have low-efficient probes or high-efficient probes. And we can have this kind of iteration more than 1,000 times for a fixed panel, for example. And this is the kind of tool that we use for our for our custom panel, for our user. So regarding our tool set, so we obviously have our fixed panel, so Exome 2, I will show you one slide on this, but also we have custom panel. We make DNA, so we make exactly what you want, whatever the omic application you would like. We have also all the reagent to perform the library prep, the enrichment. We have also specific product for cell-free DNA, so liquid biopsies. This is what Julie will show you after my presentation. We will have also all the tools to perform transcriptomic, epigenetics. We have also the possibility to adapt our protocol for long-read sequencing. This is something also that Mayra will show you after my presentation. So we have a continuous of product for this kind of workflow. So from the library prep to the end of the target enrichment. So we have this flexibility in Twist, meaning that you can use a box for a box with reagent for enzymatic fragmentation. You can use other tool for mechanical fragmentation for those who would like to have a mechanical shearing. We have also a tool for the ligation step, meaning the universal adapter system or other tool. So we have the flexibility to provide you all this kind of tool. Just now, I would like to show you a little bit the workflow itself. So the workflow, it's always the same in NGS. Our philosophy, it's 3 steps: fragmentation and repair, e-tailing, after ligation and after amplification. So if you choose to perform enzymatic fragmentation and repair e-tailing in the same step, you will enter in the ligation step. In the ligation step, you have 2 options. Okay? Either you would like to perform, for example, whole genome sequencing, PCR-free. You can use full-length adapter during this step. So it's option 1, okay, in yellow. So it means that you will ligate during this ligation. You will ligate full-length adapters that will bring indices during this step. After this, if you want to enter in the whole genome sequencing, you can load it on your sequencer. If you don't have enough field, it's always possible to amplify. Since our adapters are TrueSeq compatible, you can amplify this with P5, P7. And the other option, this is the one that is mainly used for target enrichment, it's using the stubby adapters. It's option 2. So during the ligation, we use a short adapters, which increase the conversion rate, meaning that at the end, you will amplify with more complexity your DNA. So it's mainly used for target enrichment because we want to prevent the PCR bias, obviously. So if we increase the conversion rate, you will have a nice complexity, which is quite important in NGS. A brief summary regarding the capture. Our probes are double-strand probe, biotinidine 5', so we use streptavidin bits. So meaning with your prep library, we pull them, okay, because they are all indices. We pull them, we put our blockers, we put all the hybridization reagents that brings the stringency, and then we hybridize. After some washes, you are able to load this on your sequencer. Regarding our exome portfolio, so the story began in 2018. So we had our first core exome in 2018, and now we are in 2025. We release our new CNV spiking panel because our workflow, our product has now a really disruptive exome with the update content, which is the exome 2+, which is mainly used in Europe. And this content is also used for our precision exomes, the IVDR one that we released also 1.5 years ago. So the CNV spiking panel, it's a new one. It's a new panel that has been released recently. I will show you one slide to give you some explanation regarding this. Regarding our exome, just some data. Just to let you know that the content of the exome 2+ is up to date, and what is really important for us is to have the best, the optimal result for this. So you can see on the bottom of this slide the Picard metrics recorded, the 480 base penalty, which is a metric that we really like in Twist because it shows the uniformity of capture. It's really low, so 130 for those who know these metrics. We have a nice uniformity, nice complexity, nice specificity, and the coverage, obviously. If all these three metrics are okay, it's perfect. Regarding the CNV backbone, so yes, we released recently this CNV backbone, so we designed it. So the idea it was to complement the exome solution because our exome solution, with the exome solution, you can perform CNV detection, but indeed you have a lot of gap. So the idea it was to complement the CNV backbone, and in our workflow we have the flexibility to target this intronic region with this spike-in. So we can use our this spike-in, so whatever the resolution you would like to use, 25 kb, 50 or 100 kb, we have this possibility to use this for your CNV detection. So this is 3 panels that are off the shelf today. So don't hesitate to come back to our booth to discuss all of this. So obviously this is something that have been validated. So we have a poster in the ESHG. So don't hesitate to go to this poster. I put you the number of the poster. And we have also, I saw the Genome team, but we have validated this panel with Genome, so with the software companies that validate the tool to analyze the CNV, and with SEC1 as well. Regarding the custom panel, because we can make the panel you would like, whatever the omic application you have. So indeed, we are quick, so meaning that in 2 weeks we can synthesize our probes easily. After this, we need to QC this and ship it, but it's really quick. Okay? We can make a panel for metagenomic, for viruses. You can do everything you would like. But just, it's always better to discuss about what we can do with this, with a really nice application that we set up in Montpellier hospital, for example. For example, in this one, we discussed about the possibility to have an alternative protocol for the Myriad MyChoice-DX protocol. So the idea was to internalize this protocol for the HRD status, to check the HRD status. So they had really degraded sample, so FFPE samples. They wanted to have a panel that cover all the genes that are involved in HRD, so homologous recombination deficiency. So the idea it was to work with them. So we did the first panel. We had the first version with 22 or 21 genes, something like that. The first results were good, but not enough. So we use our bioinformatic tool to set up a new version of this panel. And now we have 23 genes. All these genes are involved. You can check the BRCA status and things like that. But in HRD, okay, you know the mutation, but you need to know also what's the effect on the genome itself. So the idea it was with the same library prep to perform a custom panel, but also a shallow whole genome sequencing. So this is a possibility with our workflow. So that's what we did. So to just explain you this slide, we performed the EF version 2, the enzymatic fragmentation version 2 protocol. We perform 8 cycles, and with this one, we can do a whole genome sequencing directly on the sequencer. We can put 8 samples on an NX500, or if you want, you can perform 65 custom panel on this sequencer after target enrichment. So this is a nice slide to show you the customization of your application if needed. So we are not only involved in this kind of application, we are also involved in transcriptomics. So we have also all the product to perform whole transcriptomic or targeted sequencing. So it's something really, it's for 2 years now, 2-3 years, we have this kit. So it's something that you can use if you would like to work on fusion exomes, things like that. We have an off-the-shelf panel that have been designed in partnership with the Seagate. We have also an exome panel, but the exome panel for RNA-seq has a different design. That's what I wanted to show you, because RNA-seq, we need to place our probes on the transcript. Okay, so the idea it was to have a different strategy compared to the standard one to place our probes. And as you can see on this slide, we have really we increase the number of reads thanks to this new strategy. So it's a really nice strategy that our bioinformatics team set up. So regarding the liquid biopsy, so I will go quickly on this because Julie will give you a nice presentation regarding this kind of product. So we set up 1 year ago a protocol. We released this protocol, meaning that you can work with low input. You can work with UMI, for example, when you want to go really deep in the coverage. So this is something that we released recently, and it works perfectly thanks to a new ligase. We enter in a new era with Twist, because now we not only perform the DNA synthesis, but we also create enzyme, and our new T4 ligase is amazing. You can see here the library yield, so meaning that if you increase the library yield of your library preparation, you increase the complexity. So obviously when you go deep in sequencing, it's really interesting to have this kind of complexity. The last slide, it was to say, so as you will see with Mayra, so we are completely platform agnostic, meaning that our job is to take your DNA and to give you an enriched DNA. Whatever the sequencing platform, you are able to use it. So we have a partnership with PacBio, with Element. We are also obviously Illumina compatible, and we have also a partnership with Oxford Nanopore regarding this. So all our protocols are completely, it's completely usable for this kind of technology. And a little slide on this. For example, for the AVT element, so we have a new partnership with them, meaning that we can use all our protocol, all our tools protocol, and with this excellent machine, we can use this protocol and convert the library with this Warring cycle amplification in the sequencing itself. So you don't have to do anything; the sequencer itself will do this for you. And for those who would like to use the blockers from Element, because they want to perform a library with Element adapters, things like that, we have also this kind of tool. And the new thing is we have we collaborate for the Trinity workflow. So meaning that the Trinity is something that you can discuss with Element, but it's amazing because you will have to do only the library prep. Meaning that at the end of the library prep, you put this on the sequencer and the capture will be done in the sequencer itself. So it's perfect. So it was my last slide. So just a word to say, so come to our booth. We are here. So don't hesitate to come and ask me a question, technical question. It will be happy to reply. So the next presentation, it will be Mayra. So Mayra will give you a nice presentation on long reads. So this is what we discussed just previously. So I let Mayra coming to the stage. Thank you very much. Thank you very much. See you.
Still loading, so. But yeah, thank you, Yann, for the kind introduction. I'm very happy to be here. I'm Mayra Sauer from the MGZ Munich, and yeah, first the disclosures. So we work in a collaboration with Twist Bioscience, and we received some complementary reagents. But first of all, I would like to introduce the MGZ Munich. So what are we doing? We do clinical genetic counseling, and we are a human genetics laboratory. We are seeing patients and their families, and we're doing a comprehensive genetic diagnostics for now over 25 years. We just had our anniversary, which was very nice. And yeah, we are still growing. So last year we processed more than 40,000 samples, and we are more than 200 employees. Our expertise for sure lays in the genetic in-house consultations and by our physicians where we see the patients, but also the genetic counseling for other clinicians. And we have a very strong focus on the R&D. That's why we have an R&D department where I'm in, because we want to provide the best quality and always the best method for our patients. And what you can see here is, we have a broad portfolio on diagnostic tests, which we use for various disease fields. But today I would like to focus on one topic, which is cancer. So how do we identify variants that cause hereditary tumor syndromes? Our current method is the whole exome sequencing with short reads, what Yann introduced. And there we have a clarification rate of hereditary tumor syndromes of 25%. So the question was, okay, what is causing the other 75%? And as you all know, there are very different types of variants. I will just highlight them again. What you all know are variants in the coding regions, but there can be also variants in intronic regions or variants that affect splice sites, as well as UTRs or promoter regions. So with our current methods, the short-read whole exome sequencing, you can detect SNVs and indels in coding regions and also copy number variants. And that's why we thought, okay, maybe we should extend our analysis and use long-read sequencing to analyze our patients, because we hope that we will see intronic variants, that we maybe see variants in the UTR regions, and also structural variants. And now you could ask me, we decided we want to go with the PacBio sequencing, with the high-phase sequencing, and then you can ask me, of course, why are you not doing whole genome sequencing and long-read genomes? We had a lot of talks the last days, which actually show their diagnostic yield, the increase in the diagnostic yields, but we have much more patients, and so we wanted to have a high throughput application with more than 3,500 samples a year, and with one PacBio Revio machine, that's not feasible. So we reached out, and luckily Twist had a very nice kit, which is the target enrichment for a long read, and we decided, we want to sequence that on a Revio. Twist has other kits. They have a PTX and a dark region panel, but we wanted to study the cancer genes, so we designed a customized panel comprising of 175 cancer genes. Just to give you an idea, like the whole genome sequencing, you have a target space of 3 gigabases. If you go with a target approach, in our case, it's still a big panel. It's 14 megabases. It's still pretty small. And it is actually, to give you the fractions or an idea, we have only 7.3% of exonic regions, 1.23% of 3' UTRs and 5' UTRs, and over 90% is actually the intronic region. So we include the whole genomic information, and you can see how much that is actually for these genes. And just to scale it up, so if we go with that approach, we could process more than 30,000 samples a year, which is much more than 2,500 whole genome sequence. Now I would like to introduce the workflow or the protocol. So it starts with an extraction of the genomic DNA. This needs to be fragmented, like also for short reads, but not as short. We shear to 5 to 7 kb. We use the genome grinder because there you can shear 96 samples, even more. You can actually 96 well plates shaking in one go. And then you start with the Twist library prep, which is what you know already, comprised from end-array repair, DA tailing, and you add barcoded adapters. Then you have an overnight hybridization with biotin-labeled probes. and then you capture your targets with streptavidin magnetic beads and wash away the off targets. And then in the end, you have to amplify because otherwise it's too little. And then you start, in our case, the PacBio library prep. It's called Smart Belt Library Prep. You do the ABC and you sequence on a review. The advantages of that are that you can multiplex samples, that you still have a high coverage or higher than a whole genome sequencing run. You have a high accuracy because we use the hi-fi reads, and because of the long reads, you are able to do haplotype phasing. As I said before, we work with a customized panel. Of course, you can do whatever you want, and it's much more cost-effective than the whole genome. What you don't see is actually methylation because you have different PCR steps in the library prep. That's why you lose the information about the methylation. Now I would like to show you or compare the two methods. Here up there you see the whole exome sequencing data in the IGV. Here you see our targeted long read method. We have a homologous coverage over the whole gene. You can also cover GC-rich regions. And as I said before, you will also get the phasing information. We have a higher median coverage, and in our case, we multiplex 24 samples. These are QC data from our PacBio run. So usually what you have with short reads is a Q30, and you have 150 to 300 bases. In our case, you have a much higher Q score. It's the mean is Q49, and of course you have longer reads. This is of course what we want, so it's just what you see. And now we were tackling the question, do we really see all the variants we were hoping to see? And this is what I would like to show you now. So yes, SNVs is easy detectable. I mean, here you see the whole exome. Here you see I phased it already, the SNV. Also small indels are easy detectable with both methods. But what you also see are insertions, like here's an insertion of 130 bases. It is in an intronic region, so we didn't saw that with a whole exome, but of course with the long reads. And now the question was, okay, how is it with larger deletions? And because we have a targeted space, it means we really target certain genes. And there are three different possibilities. So you can have a deletion where both breaking points are inside of the target. This should be easy detectable, but we want to see that. But what happens when you have one breakpoint actually in your target and the other one is not? So it's like in the gene next to it, but you don't target that. So how does it look like? Or what happens actually if the deletion spans the whole gene, so your whole target, but the breaking points are outside of that? Can we actually detect that as structural variants with that long read method? So we started with a smaller deletion, which is 300 bases, and this was clearly detected and was also called. Then we wanted to see, is that also possible with larger deletions? And here you see one which is over 5 kb long. We had actually one read which spanned the whole thing, but you can also hear the clear coverage drop. And we used structure variant callers, which are called PBSV and Sawfish, and they both called it as a clear deletion and could tell us this is over 5 kb long. So if both breaking points are inside of the target, that's pretty easy. How does it look like with only one breakpoint? There it was more difficult, for sure. You see here a clear breaking end, and you have a lot of clipped reads in the IGV, but still the callers could call there is a break end, and if you then aligned, because you have the long reads, you align these clipped reads to the reference, it will actually tell you exactly where it is and where the second point is. So this was also quite nice, but then we thought, okay, how is it if the breaking points are not inside of the target? And this is much more difficult, what we also expected, because it's just the target is gone, kind of, but how do you detect that? And the structure variant callers couldn't see that, but in the data you could actually see the drop in the coverage, So what you would need, or what you need here for this case, is that you need a coverage-based algorithm, like it is mostly implemented for the whole exome sequencing, and you have to compare it to a negative cohort. There you clearly see the difference. And this is also, or this coverage information you also need for a duplication, because this was also not, in the beginning, not really called from the structure variant callers. But in the data, you could clearly see that there is an increase in the coverage, and you could also use the phasing information, because then you saw, okay, one allele is just double than the other, which was quite nice. But there, yeah, we need to adapt the callers. And then we asked our question, how is it with homologous regions? So in our target space, we have the PMS2 gene. It has a very homologous region, or has a pseudogene in these exons. And so we know that we had patients where we had variants in these exons, but with a short read sequencing, we couldn't distinguish between the pseudogene and the PMS2 gene. So we were thinking, okay, how can we analyze that? And we did this in a collaboration with NGenome. And in the end, it was actually quite nice. So we had 16 SNPs, or SNVs, 4 indels and 1 deletion. The deletion was not called in the short read sequencing data, and all the variants were called, but you couldn't distinguish between the pseudogene and the gene. With the long read data, you could actually quite nicely distinguish between the pseudogene and the gene. And this is what you see also here. You see the 4 haplotypes and clearly the variants. So the long read method was able to do that. And if you want to get more information, please visit the poster or talk to them. Yeah. So in summary, we can say that target gene enrichment with the HiFi sequencing can combine the advantages of the HiFi sequencing technology and the capture enrichment. It's much more cost efficient, and you can see actually structure variants in non-coding regions. It is possible to resolve the homologous regions, but we still need a good tertiary analysis exactly for the large structural variants where the breaking points are outside of the target. And yeah, with that, I would like to thank you for your attention, and of course the whole team who was working on the project, and yeah, our collaboration partners Twist and PacBio and Beckman. Thank you.
The next one will be Julie. So Julie Vendrell, who come from Montpellier.
So hello everyone. I'm very happy to be here today to share with you the reflection we had before to implement the ctDNA NGS testing in our lab, and to present the workflow we developed for this application. So just in a few words, cell-free DNA is short fragments of DNA that are released in the bloodstream by dying cells. We estimate that around 300 billion cells die per day. And when we analyze the composition of the cell-free DNA, we realize that 60% comes from hematopoietic cells, and around 10 or 20% comes from vascular epithelial cells. So not all the cells release in the bloodstream, or DNA is stable in the bloodstream. After, it's only small amount of DNA that you can have. It's around 1 or 5 nanograms, which represent among 1,000 genome equivalent per ml of plasma. These amounts can vary, depends on physiological or pathological conditions such as aging, activity, physical activity, inflammation, trauma, or disease. The half-life of cell-free DNA, it's around 15 to 100 minutes. It's several studies estimate at that time. So we can say that liquid biopsy, it's a snapshot of cell-free DNA at a specific moment in time. In case of cancer, we speak about circulating tumor DNA or ctDNA, and ctDNA represents only a small fraction of the cell-free DNA. The advantage of liquid biopsy, aside from the fact that it's less invasive, of course, than tissue biopsy, is that it reflects the tumoral spatial heterogeneity. As DNA is shed from all primary site and metastatic sites. It's a very powerful tool that now it's well accepted and used by oncologists, and the oncologist can ask for this analysis at different steps of the patient care pathway. So the first one, of course, is at diagnosis. It's when the tumor sample is not accessible, or DNA is not analyzable, or if there is no remaining material after immunostaining anatomopathologist experience. The other step would be at monitoring. You can follow specific alteration and look at response to treatment, or identify earlier relapse, or at the relapse to identify a mechanism of resistance. Circulating amount depends of cancer type, tumor stage, and also how and when sampling have been collected. So problem with cancer type, there is some pathology where there is a lot of circulating ctDNA, like for example in colorectal cancer, the tumoral fraction is around 20 to 50%, so it could be easier to detect alteration. But for lung cancer, where there is the most application, it's most around 1 to 5% of ctDNA in the cell-free DNA. So now in the international guidelines are supported the use of ctDNA to take patient care, but if results are negative, the tumor testing remains the gold standard. But we need to have very sensitive method to detection. So actually there is two commonly used in routine. So it's ddPCR and NGS. Of course, all of them have advantage and disadvantage. So ddPCR is very sensitive, high throughput, and with a low turnaround time, but it's very limited to the number of alterations we can analyze per samples. On the opposite, NGS allow large panel detection alterations. There is some commercialized solution that we will speak later, but the sensitivity is lowest. We need to use UMI. The problem that there is no bioinformatic pipeline available for free, actually, and there is a high turnaround time, and the cost, of course, is most efficient, and it needs some high-capacity sequencer. And not all the hospitals have this kind of sequencer. So before to implement the technology in the lab, we had an overview of what was available on the market. And so this is just a representation. There is other companies who propose these kind of things. But in all the case, it was integrated solutions. Because as I told you, they were not available bioinformatic pipeline to take into account the UMI and this kind of application. So the two first, you have also to send the samples to the company, and the other one are huge, large panel, comprehensive genomic profiling panels that required high-capacity sequencer. The problem we have here is in the lab we don't like too much to send samples or data to outside the hospital, so it's not solutions that we really like. And furthermore, we don't have the sequencer. And finally, the limit of detection for us was a little bit a limiting factor because they have a limit of detection at 0.5. And when we had a look on the results we obtained with ddPCR, this is a result from more than 2,000 non-small cell lung cancer samples that we analyzed in the lab by ddPCR, we detected more than 600 alterations. And here you have the representation of the VAF, the variant allele frequency for the positive alteration detected. And you could see that if we have a limit of detection at 0.5, you will miss almost 40% of samples, and you will deliver a negative results for these samples. The other point, it's that we said this is modelization, so we know that the probability to detect a variant with at least 3 altered reads can be modeled by a binomial law. And this probability for a specific VAF is function of the depth of coverage. So it's why it's represented on this graph. So in other words, if you have a depth of coverage of 2000x, you will have 99% of chance to detect an alteration at 0.5. But with the same coverage, you will have only 50% of chance to detect a variant with a VAF of 0.2. And if you want to detect a variant at 0.1, you will need at least 9,000, more than 9,000x. So after all this assessment, we decide to develop our own workflow using Twist as a wet part of the process, and to develop our own bioinformatics process. So for Twist, Yann already presents the process, except that in this application we don't need to do a fragmentation because DNA are already fragmented. And obviously we need to introduce some UMI to reduce PCR bias and to increase the sensitivity to quantify altered alteration. So after the process is the same that we saw before. So our panel, we developed a panel that could take into account all the samples we have to deal with in the lab. So the panel is very practical. We have only the genes for which there is an oncologic activity or that are targetable. So it's around 300 probes, 162 targeted regions that correspond to 33 genes, and it's around 30 kb. If you feel free to contact us if you want to share, we can share the panel with you, it's not a problem. With this panel, we can analyze around 10 to 12 samples on a mid-output Nexus 500, that is the sequencer we have in the lab. So just a word on our bioinformatics pipeline. I'm not the best people to speak about that, but I will try. So first of all, the first module is UME-based deduplications. So you align all the reads. We identify the reads that have the same UMI to generate a consensus UMI, consensus reads. And after, with all these consensus reads, we align and do the variant pooling and annotation. In parallel, there is a quality module. So it's basic, it's multi-QC, fast QC. And after, we combine both on a reporting. It's not a reporting, it's an interface, graphical interface that I will show you. So the pipeline is open source. You can download it on GitHub. So the same, feel free to download, to use, and to come back to us if you have some question to use it. So the specificity we integrate in this pipeline, it's a dynamic limit of detection according to the depth coverage and the input of DNA we use. So like that, we can be more sensitive for samples that we have a high quality. And we also use a whitelist. So it's clinically relevant alterations that we extract from the OncoKB. So it's all the alteration present and that are also in our panel. And we also enrich this list by our own alteration we detect on tissue samples. We also have a blacklist with known artifacts that we can remove from the analysis to avoid some noise. So the dynamic LOD was possible due to the panel uniformity that we have. So that's something Yann also explained you. So on this graph, we have ordering the coverage of all the target region of our panel from the lowest to the highest covered. And as you could see, it's very uniform, and more than 90% of the probe have a coverage between mean, present, and standard deviation. So it's why we could apply to each sample a specific limit of detection. So just to show you the performance of our workflow. So for that, we use some positive controls that we order, and that contains very relevant alterations that we need to find in a clinical routine. And they are well characterized by ddPCR. So on this part, it's a positive control with alteration at 1% that are here, and you saw a very good correlation between the variant allele frequency observed and the one expected. When we use a positive control at 0.1%, here we find we can miss some alterations. So this one we missed in all the in both experiments we've done, and this one we missed in one of two. But it's normal because on these samples I forgot to put the LOD, but we are around for one experiment at 0.3 and for the other one at 0.4. So it's not, as I showed you before, it's not normal, but we can miss it. Now if we also analyze the reproducibility intra-run by with different DNA input, amount of DNA input. So if we work with 20 here, if you work with positive control with VAF at 1%, you have no problem to detect, even if you are working with 20 or 10 nanogram. You could see here the LOD. Of course, it also depends of the input you use. But now if you are working with the control at 0.1, we miss some alteration when we use the 10 nanogram of input. But once again, it's normal. As for the sample, the LOD was at 0.5. After we tried to use different sequencer and we analyze the same library on an AVT. So as Yann said, no problem, without nothing to do. And even with the bioinformatics pipeline, the results were straightforward. After I want to show you that performance of the process with samples that we analyze. So among all the alteration detected, 155 was on the tissue samples, so it's true positive. But we had, that's the interface. So when the variant is in the white list, it appears in blue, as you can see. If they are not in the white list, they appear in green. And here you have a summary of the limit of detection, average, and deduplication ratio. So that's also some other true positive. So if the variants are in the white list, they appear even if they are lower of the limit of detection. So that's very good because you can detect like that some alteration very relevant for patient care that are under the limit of detection. We also, this is a false negative, so it's samples. It's alteration that we saw by ddPCR, but we could not detect by NGS. But once again, they are very low and they are under the limit of detection of our NGS. So it's for that easier to deliver a diagnosis, and we can say, okay, we didn't detect at this limit of detection. And the other one, they were not detected. They were detected in the tissue samples, but not detected by ddPCR, neither by NGS. So perhaps the samples was not relevant, or the tumor samples, the ctDNA was not present in the bloodstream. So regarding the false positive we detect, we had 17. These false positives are not, we consider that false positive for the moment because they were not detected in the tissue samples, but some were validated by ddPCR, so they were in the liquid biopsy, so it really reflects the tumor heterogeneity. One was not detected by ddPCR, so perhaps this one is a real false positive. And then 10 are under investigation. We are designing the new ddPCR probes to see if they are or not false negative. So in conclusion, I will not come back on the clinical utility of ctDNA analysis, but perhaps I hope I show you the importance to have a limit of detection that can vary depending on the amount of samples, quality of the samples, quality of your technique, of course. We develop an open source bioinformatics pipeline that is, as I told you, available on GitHub, and that will be coming soon on NFCore. And we now have implemented these techniques in the lab for routine diagnostic use. Just to remind, liquid biopsy is less sensitive than tumor tissue-based genotyping. That remains the gold standard, but in all the cases, we need to propose performance process to detect this kind of alteration. So the one we propose here have a good sensitivity and precision compared to ddPCR. I would like to thank all the persons who works on the project, from the labs, the clinicians, and the bioinformatics teams from Montpellier who participate to the development of the bioinformatics pipeline. Thank you very much.
So thank you very much, Julie. So it's the end of the seminar. So now we are in the Q&A session. So you have some mic. If you want to stand and ask your question, feel free.
Hi, Anirudh, clinical geneticist from Sweden. I have a question to Mayra. I was wondering what realistic diagnostic yield are you expecting from this panel?
This is a good question. Actually, we don't know. Yesterday there was a talk where they compared like whole genome sequencing with short read whole genome sequence, and they saw a diagnostic yield increase of 10 to 15%. So I don't think that we will have so much, but at least what we see already is that we can find a second variant which can then make it more plausible and in the intronic regions where we have a lot of patients where we think there is something, there has to be something because the phenotype is so clear, but we can only find one variant, and this doesn't explain everything. So we don't know yet. We don't have the data yet, but yeah.
So the interesting thing is like you have to realistically compare it to the same panel for a short read sequencer. Then you'd like it would be much cheaper.
Yes,
and like intronic variants could be found in any other sequencing technology. It's like very complex variants, probably the long read is better, but then like for the normal variants, it's just about the probes.
Of course, of course. But I think also with the phasing information, you get more, you can, as I said, like differentiate between the pseudogenes, and so it will give more information. But we don't know yet.
Yeah, but there are not many recessive genes in hereditary cancers.
That's true. You're right.
Thank you. Any other question? Yeah. Good.
Hi, Gaetan Biou from Medical Genetics. Thank you very much for your talk. Both were very great. I have a question more on the technical part. Were you curious to sequence before enrichment and after enrichment of your long reads to see whether there was any bias towards potentially destroying some very long reads, right, through the capture? Any size bias or any potential artifacts, which I'm thinking oxidative C2T transition that might happen more on those really long reads after enrichment?
You mean if we have a bias towards longer?
That would be towards shorter.
So the technique actually like there is like a cutoff. So I mean, you fragment your DNA to a certain size, so the fragments are already small, long, like 5 to 7 kb. But then also, if you want to get, or we saw that with the kids, if you want to enrich and have a very on-target rate, there is a bias towards smaller, like 3-4 kb fragments. So. So we actually see a better coverage and with the shorter long reads than that we have too much long reads which are just giving us off target. So yeah.
Besides the size, any C to T transition due to the oxidation?
Not yet.
Not yet.
But
thank you.
It's a good idea to look at that.
Yep. A question for Mayra or for the Twist staff. So do you think that your approach can be translated to the clinical exome? So it is possible to create, let's say, a panel, including the homing gene plus the introns for diagnostic purposes?
The question is, how big is it then in the end, right? If you want, like, I mean, you saw the fractions, right? We have only 7% of exonic regions and over 90% of intronic. Just for these 175 genes, it's already 14 megabase. If you do the whole exome, which is bigger, about 40 megabase, plus the intronic, then I think you...
Yes, but it's shorter than
genome. It's a genome, that's the thing. Then you can go for a genome.
Yes.
So.
But definitely you can do a custom panel, obviously, with Twist.
This is the thing, you can do... Okay. You can ask
this is possible, obviously.
Are you planning to do this? Are you planning to create this
we do what you want. So if you want something like that, it's possible.
It's not for me. Probably it's for all the people.
We can discuss afterward if you want. Sure. Any other question? On my side, I have one question for usually regarding the input. So you work with between 10 and 20 nanogram. Did you try to work with less than 10 nanogram in the cell-free DNA workflow?
Yes, but with tumor samples of patient tumor samples. Actually, we try to do our best for the samples we receive. So it's why we did this analysis.
And the result?
We find some alteration, targetable alterations, but due to with the dynamic limit of detection, it's easy because and also the whitelist because even if you find an alteration.
What was the less amount of DNA that you tried?
I think it's 4 for the moment. For the moment, 4 nanogram.
Okay. Great.
Yeah.
Great sensitivity. Perfect. If any.
But just if for the moment, because we did only 8 runs of diagnosis, so each alteration we found, we validated by ddPCR for the moment before, just to be sure and to know the limit of our pipeline. But it's time to get used with these new tools, and after that, I think we take confidence in our tools, and we will stop. But for 4 nanogram, we validate it.
Great. Well, thank you very much. I think we are out of time. Yeah. Thank you very much for this. See you at our booth when you want. Thank you. Bye.